My name is Emily Nan, and my partner is Daisy Cortez. And our project was on spheroid compaction as a marker of metastasis. And so, specifically, we wanted to look into the effects of the CSRP1 knockdown and stem cell media in non small lung cancer. So, lung cancer has the highest mortality rate between all the cancers, and this is due to its high metastatic potential. So, when I talk about metastasis or metastatic potential, it just means that the cancer is able to travel from its primary site to its secondary site. And the issue with this is that lung cancer can't be detected till it reaches its fourth stage. So, by this point, it has already moved to the rest of the body. And studies have shown that cancer patients in general have significantly lower survival rates than the patients without this mutation. And that's why we want to look into adhesomes. And adhesomes is just a structure of proteins that connect to each other, and this allows cell to cell or cell to ECM interactions. And our specific protein of interest is the CRP protein. And that's because this is the protein that matches with the CSRP1 gene. And then preliminary in vivo work from our lab shows how there's an increase in metastatic aggression when you knock down the CSRP1 adhesome gene, in addition to growth in stem cell media. And the difference between these two medias is just that the stem cell media is enriched with nutrients and stem cell factors to mimic the cell's natural environment. And these promote low cell death and high proliferation. And looking at past literature, you can also see that the Hayes cell line has a high compaction while also displaying higher invasion than the non-compact cell line.
Now, let's take a look at our hypothesis and objective. We hypothesized that knocking down CSRP1 combined with stem cell media would increase spheroid compaction, suggesting a greater metastatic potential. Our objective was to determine whether disrupting the adhesome through CSRP1 knockdown induces spheroid compaction in tumor spheroid models derived from various non-small cell lung cancer cell lines under both regular and stem cell enriched medias. To test this, we cultured spheroids using four non-small cell lung cancer cell lines, A549, H1299, H460, and Cal-3. We monitored them over X amount of days depending on the cell line and measured spheroid area and circularity, two indicators of compaction and by extension metastatic behavior. In the A549 cells, we observed that CSRP1 knockdown combined with stem cell media significantly increased compaction. Interestingly, in the H1299 cells, CSRP1 knockdown alone was sufficient to drive this effect. These results were reflected by decreases in spheroid area and an increase in circularity. We saw consistent trends in the H460 and Cal-3 lines. The combination of knockdown and stem cell media led to the highest degree of spheroid compaction, further supporting our hypothesis across multiple non-small cell lung cancer models. So, what does this all mean? Our results suggest that adhesome disruption and the tumor microenvironment work together to drive spheroid compaction, a potential physical marker of metastatic potential. Moving forward, we plan to explore other adhesome genes to better understand their role in non-small cell lung cancer progression and identify new therapeutic targets. Thank you so much for watching.